Hello, Dr. G. For the video assignment, our topic will be on virus-like particle or VLP based vaccine. So VLP represent one of the most exciting emerging vaccine technologies for generating effective and long-lasting protection. As for introduction, I will be explaining about what is a recombinant vaccine before we'll move on to VLP based recombinant vaccine. Recombinant vaccines are produced by inserting the DNA encoding an antigen that stimulates the immune response into bacterial or mammalian cells, expressing the antigen in these cells and then purifying it from them. There are three types of recombinant vaccines: subunit vaccine, which consists of components of pathogenic organisms, including proteins, peptides, and DNA; attenuated vaccine, which are genetically modified pathogenic organisms that are made non-pathogenic and used as vaccine; and lastly, vector recombinant vaccine. They are genetically modified viral vectors that can be used as vaccines against certain pathogens. Basically, recombinant vaccine is produced using recombinant DNA technology method, where we can isolate and clone single copy of a gene into an indefinite number of copies. First, a restriction enzyme is used to cut the DNA molecule in the host plasmid, bearing the desirable gene sequence, resulting in a linear molecule with single-stranded sticky ends. Then annealing occurs where ligase enzyme join the vector DNA with the gene of interest, producing a recombinant DNA. Last but not least, insertion occurs where recombinant DNA is inserted into a new host cell. Replication of the cell carrying recombinant DNA molecule occurs, producing genetically identical cells population, or we call as clone. These are the latest recombinant vaccine developed mainly in 2017. First is a Zika vaccine targeting NS1 protein, protecting the immunocompetent adult mice, mainly to boost the humoral and also cellular immune responses. Adenovirus recombinant vaccine carrying toll-like receptor 5 for cancer immunotherapy. Malaria vaccine, chikungunya fever vaccine utilizing an insect-specific virus platform. And lastly, engineered trivalent immunogen adjuvanted with a sting agonist, mainly to protect against Trypanosoma cruzi infection.
What is virus-like particles? Virus-like particles, or VLP, is a multi-protein structure which are constructed of several identical protein copies and are able to self-assemble into capsids or non-enveloped capsids. Sizes of VLP ranges from 30 to 90 nanometer. It has a similar biochemical composition, morphology, and sizes with the authentic native viruses. However, VLP lacks of viral genome, which makes them non-infectious viral particles. Hence, VLP are able to be served as a nanocontainer. The application of VLP. VLP are able to be used as a vaccine, vehicles in drugs and gene delivery, and as tools in nanobiotechnology. The role of VLP-based vaccine will be discussed more throughout this particular discussion. While the function of VLP in nanobiotechnology and nanomedicine will ensure the effectiveness of medicine or to enhance the function of materials such as dyes or enzymes. Why VLP is used as a vaccine? VLP are used as a vaccine as it exhibits traditional vaccine parameter. The immune system are able to recognize it as it has the exact properties as a native virus, and an immune response are able to be triggered. VLP-based vaccine are also safe to be used as it is unable to replicate. In other terms, it's not infectious anymore. Broad host spectrum of VLP means that VLP are able to be constructed via recombinant technology by using different hosts, whichever is suitable. Flexibility, which means that rapid and efficiently produced. Hence, rapid immunization are able to take place especially during emergence of novel epidemic or pandemic flu strains. Versatile properties and indication that VLP are able to be modified based on the application. For example, multivalent vaccine are able to provide protection against 9 different strains of HBV. Stability and uniformity of VLP shows that VLP are resistant or closely resistant to changing its property, hence it is easier to be controlled. Mechanism of VLP as vaccine. Since VLP-based vaccine resembles the original native virion in terms of biochemical composition and sizes, so it has the properties to self-assemble into lipid-enveloped or non-enveloped capsids. These particular vaccines are injected into the veins and eventually traffic to the lymph nodes, where most of the white blood cells or immune response are located. When that happens, more cross-linking of B cell receptor with VLP occurs, as VLP are able to display multiply epitopes in repetitive and dense array, which enhance the B cell activation and response. The unique repetitive surface structure formation also enhance T cell stimulation and cellular mediated immunity. So basically, VLPs triggers a high humoral and cellular immune response, as VLP-based vaccine are efficiently recognized by APCs or antigen-presenting cells due to the repetitive structure or subunits without causing any harms to the human body. Characteristic and techniques for VLP to be favorable for vaccine development. First, the small sizes of VLP makes it optimal for rapid drainage to the lymph nodes, which also means faster immune response are able to be triggered. Second, modifying the exterior facets of VLPs for effective B and T cells responses. For example, the target antigen are able to be displayed to B cells in authentic and native configuration by using multiply methods to modify the exterior facets of VLP, either by using chemical or genetic fusion techniques. The chemical techniques for antigen display on VLP are mainly based on modifying one of the following chemical groups on the surface of VLP: amino groups, carboxylic groups, or hydroxyl groups. Lysines and amino acid residues on the surface of VLPs are the most commonly used target for modification. Genetic fusion techniques, on the other hand, are considered powerful tools in manipulating the exterior surface of VLPs, while chemical techniques do not always result in full modification of all monomers building up the VLPs, in particular if large antigens are to be displayed. The genetic techniques are mainly based on two methods, namely direct introduction of peptides epitopes or manipulating of the structural viral genes for more efficient introduction of desired peptides. The interior facets of VLPs are also exploited for effective delivery of adjuvants. VLP lack of genome, but many are capable of packaging nucleic acids during expression process in the host. Such naturally packaged nucleic acid can be enzymatically digested from the interior facets of VLPs, leaving a positively charged inner surface, which then can be successfully decorated with a range of different other adjuvants. Adjuvants formulation. Although VLP-based vaccines are generally highly immunogenic, formulation with adjuvants tends to enhance the magnitude, quality, and longevity of the elicited immunity. Generally, VLP-based vaccines are formulated with aluminum salts-based adjuvants. By understanding the interaction between pathogens, an immune system leads to development of specific adjuvants. What is actually adjuvants? Adjuvants are immunomodulatory substances that are admixed with vaccine to enhance their immunogenicity, potency, and efficacy. The advantages of adjuvants formulation are: it enhances immunogenicity, broadening induction of immune response, reduce vaccine dosage, reduce vaccination costs by antigen sparing, accelerate immune response, and stimulate stronger immunological memory. Comparison of different production platforms of VLP as vaccine. VLP-based vaccines are broad host spectrum, which means it's able to use different types of systems such as bacterial system, yeast, insect cells, mammalian cells, and plant cells. Almost 30% of VLPs are produced in bacterial systems, mainly E. coli, where the genes of viral structural proteins are codon optimized for bacteria and cloned into commercial plasmids under strong promoters. This process ensures high production of the desired recombinant proteins. Up to this point, we know that VLP are able to be produced by using various types of hosts using the recombinant technology. The expression system chosen for VLP production must take into consideration the requirements for protein folding and post-translational modification. Several expression systems are available, and the main advantage and disadvantage of each system are summarized into the following table. The table shows the comparison of different production platforms of VLP as vaccine. There are initially 5 production platforms, but in this table there's only 3 type of platform that are summarized. Examples of commercialized VLP vaccines. Several VLP-based vaccines are commercially available, including vaccines against human papillomavirus, which is HPV, such as Cervarix and Gardasil, while GlaxoSmithKline and Recombivax HB are vaccines against hepatitis B virus. In addition, the first licensed malaria VLP-based vaccine, which is Mosquirix, has been recently approved by the European regulators.
Mainly, the development of efficient VLP vaccines will require the combination of diverse strategies such as different delivery system, or we call as adjuvants, to present the antigen in a manner that can elicit an adequate and efficient immune response against these antigens. The use of novel biotechnological tools has provided a new arsenal of strategies and possibilities to the field of vaccinology. In the future, we may develop a better VLP vaccine delivery methods that may allow mass vaccination system. We can also develop vaccine for non-infectious agent. We can express or foreign proteins in plants, can develop edible VLP vaccines, and also can be used to identify and utilize a better immunogen as new vaccines for diseases. In conclusion, VLP-based vaccine is one of the latest type of recombinant vaccine that has been used. VLP and its self-assembling proteinaceous nanoparticles are powerful platforms to present antigens to the humoral immune system. These platforms represent a geometrically rigid repetitive array of antigens that are capable of robustly triggering both humoral and cellular immune responses. In addition to the highly successful current VLP-based vaccines against HPVs, many other VLP or nanoparticle-based vaccine candidates have recently entered preclinical and clinical trials. VLPs have been also engineered to present epitopes from tumor-associated antigens as therapeutic cancer vaccines. The application of VLP, its mechanism of VLP as vaccine, and the formulation of adjuvants has also been discussed previously. Several VLP-based vaccines are commercially available, including vaccines against HPV and a newly approved malaria VLP-based vaccines. The potential applications of this new class of vaccine platform are very broad and seem to serve as an important next generation of vaccine, as been discussed previously. That is all for our presentation of our topic. The next 2 slides are the references, and thank you.